Good morning, everyone. I'm Simran Da Costa, and on behalf of Alton Technologies, I'd like to welcome you all for one more very interesting session, which is part of our Women's Day special webinar series. So, today's session is entitled as Perspectives of Next Generation Sequencing and High Throughput Genotyping Technologies for Sustainable Agriculture. So, a little bit about what sustainable agriculture is. So, sustainable agriculture mainly deals with farming that meets the needs of existing and future generations while also ensuring profitability, environmental health, social and economic equity. It encourages methods that mimic those found in nature to maintain soil fertility, prevent water pollution, and protect diversity. And there are various ways of doing this, like crop rotation, agroforestry, pest management, soil management, water conservation, etc. Now, in addition to this, there is one more way of contributing towards agricultural sustainability, and that is NGS, which is next generation sequencing, which has greatly aided crop improvement. So, NGS technologies they bring novel tools and concepts that can enhance the precision and efficiency of plant breeding. We all know that it has become a very powerful tool to produce genomic data, and several other integrated approaches have greatly increased and improved our understanding of the molecular systems of living things and many more. So let's understand the perspective of next generation sequencing and high throughput genotyping technologies for sustainable agriculture from our speaker of the day. So it gives me immense pleasure to introduce Dr. Nisha Singh. Dr. Nisha Singh is an assistant professor at the Department of Bioinformatics from Gujarat Biotechnology University, Gandhinagar, Gujarat. Dr. Singh holds an MSc in bioinformatics from Bhubaneswar. She has a PhD in biotechnology and bioinformatics from National Institute for Plant Biotechnology, IARI, New Delhi. She has obtained her postdoctoral from Cornell University, New York, USA. Dr. Singh has also served as a DST Inspire faculty at NIPB New Delhi from the years 2020 to 2022. Her research interests lie in genomics, proteomics, and bioinformatics. She is particularly interested in developing cost effective, high throughput genotyping technologies for genetic and molecular breeding applications in diverse species. Additionally, Dr. Singh is researching how we can apply biomarkers to diverse crops and develop new avenues to evaluate variations at the protein level. Dr. Singh has developed the first patented genic 50K SNP genotyping chip, OSSNPNKS, a single copy gene based SNP genotyping chip for genetics and molecular breeding applications in rice. Dr. Singh has developed the world's first haplotype map, HapMap1 of T to facilitate the understanding of T genome evaluation in T metabolite pathways. She has developed several genomics and bioinformatics tools and pipelines as well. Dr. Singh has worked in several international genome sequencing projects, including wild rice, wheat, mango, jute, pigeon pea, and tea. She has published more than 69 international research publications in reputed journals with an H index that is equal to 21. She has three patents and two books that have more than 1900 citations. She is an active member of many international scientific societies and organizations. She is the member of the American Society of Plant Biology, USA. She has served as a reviewer for several prestigious international journals, including Nature, Springer, Elsevier, BMC, Genomics, Bioinformatics, Frontier in Plant Science, MDPI, Journal of Food Science and Technology, Omics Group, PLOS ONE. For her scientific contribution, she has gained several prestigious awards and honors, such as INSA Medal for Young Scientist Award 2020 from Indian National Science Academy, New Delhi, India, National Environmental Science Academy 2022, Scientist of the Year Award, and NASA Young Scientist Platinum Jubilee Award 2022, India. Now, without taking much time, I would like to hand over the platform to our expert. So, up to you, ma'am.
Thank you so much, Simran, for this wonderful introduction. And I'm really delighted to uh, talking about this special occasion. So first, I would like to wish a very happy International Women's Day to all the lovely women who is working in any area, in any sector, whether it's biotechnology, science, engineering, or any domain. So, or each and every woman. So, a very happy Women's Day, first of all. So, today I will be talking about the perspective of next generation sequencing and high throughput genotyping technologies for sustainable agriculture system. These days we are talking much about the sustainable, and Simran has already uh, give a light about the what is sustainable system and what are the all these aspects. So I'm not discussing that. So first I would like to tell that my area of research in which currently I'm working on, and I'm a founder of multiomics research lab. So currently I will be working on the sorry yeah plant abiotic and biotic stress, plant genomics, nutrigenomics, proteomics, metabolomics, bioinformatics, and machine learning. So these are the area in which my groups are working, and we are mainly addressing the question related to the biotic and abiotic stresses in plant and also the nutritional aspect. So the major problem right now is that hidden hunger, that malnutrition, especially in the developing countries, because we are eating something but we are not getting adequate food and balanced diet, which we call it. So for that we need to increase our protein plates, right? And for that the legumes is really key. Earlier the research were more on the staple diet, especially the rice and wheat, but now people are shifting towards the legume crops, so that we can have a balance of both, dal and rice. So that's the thing. So we can address the malnourishment problem, and again it's a goal of sustainable SDN2. So that's the area in which we are working right now. So doing that, what are the different challenges in the crop production which I'm talking about? So as we all know that our global population is increasing rapidly, and it is expected that by 2050 it will be reaching around 10 billion. So, to meet the projected demand of this rising population, we need a sustainable agriculture system. And apart from that, there are several other constraints like carbon, polymers, our environmental factors. We have adverse climate changes in the several places, including drought, salinity, sodicity, and humidity. So these are the things which are hampering the crop production. And if I talk about the more on genetic side, so we are left with a very narrow genetic composition in elite cultivated variety, because due to domestication event we have lost our genes which are more climate resilient. So we want to bring them back so that we have a superior variety for climate resilient and to meet the projected demand of this rising population. So how we can achieve all those goals? So for that we need a functional genomics tool, because if we do with the conventional way it will take a longer time, and we don't have, because our population is increasing rapidly. So we have to enhance and increase our science at least 4 to 5 fold so that we can able to chase the goal. So my areas revolves around the genomics aspect, mainly on the functional genomics. So I will start with the very basics, that what is genomics, and how I have done in the genomics and different aspects of genomics. So all know that the genome was a term coined by the Hans Winkler in 1920 to define the complete set of genetic information present in an organism, and the study of the entire structure and function of the genome of any living organism is known as genomics. So there are two type of genomics: prokaryotic genomics and eukaryotic genomics. Prokaryotic genome is pretty straightforward, but in eukaryotic we are having additional genomes. And in eukaryotes also, like in human and livestock, if we compare the plants, so plant genomes are much more complex, because in that we are having additional genomes with this chloroplast. Because in the livestock and human genomes we are having this mitochondrial and nuclear genome, but in the plant we have to deal with the chloroplast genome. And also the ploidy level is quite diverse. Heterozygosity level is also quite high in the plant genome, because in plants we are having, wheat is, let's say, hexaploid, tetraploid. So we have a different kinds of ploidy in the plant genome. So that, and also many repeat elements, and there are no any good software available to that can tackle the genome sequence of plants. So that these are the challenges in the plant genome. So we, our group is trying to address such kind of issues through the bioinformatics pipeline. So these are the subfields of genomics: structural genomics, comparative genomics, mutational genomics, and functional genomics. And our research revolves around all the four subfields of genomics. So what are the goals of genomics? So in the goal, first we have to choose, select a plant which we want to sequence, and why we want to sequence that particular plant. And now the concept is not for the single plant. We are doing the pan genome kind of things. We are taking the representative and capturing the diversity of the genome. And after genome sequencing, we have a jigsaw puzzle like thing. So we have to assemble the contigs, because initially we have a contig. So we have to assemble the contigs, and in the assembly process, that the contigs make the scaffold. And the scaffold we need to polish, because we have a gap in between the scaffolds. So we have to polish the scaffolds. And after the scaffold we need to perform the annotation, like what are the function of that particular genes which is present, what are their proteins they are encoding for. And then we have a huge set of data that we can deposit it, and we can create a genome databases. So you can see there is a dedicated database for certain crops, other than the NCBI, dedicated database for the rice, wheat, cotton, pigeon pea, because they have the use information, and we need a good database so that we can put all the information, and it will be useful for the other researchers to work on that particular crop and area. And then we can perform the gene identification in which we are interested in, and that we can further we can use for the marker assisted selection through the SSR, ESTs, and SNPs. We can mine the marker for the trait mapping, and identifying the genes and SNPs, causal genes, we can go for the genome editing or the marker assisted selection for, in the breeding program, and we can develop a superior crop variety. So the plant genome sequence. So the first plant genome was Arabidopsis thaliana, which was we sequenced in 2000, and it is a pretty small genome, 125 MB, total 25,000 genes present in that. And later 5 year we have a first draft of rice genome, and it's a gold standard rice genome. And India was the part of this rice genome sequencing project, and it has a 389 MB genome size, total 36,000 genes present in that. And later on the pigeon pea genome came, and it is completely indigenous. The Indian scientists have decoded the pigeon pea genome, and this was the first legume genome sequenced by the Indian scientist from my PhD lab, and we were the part of this genome sequencing project. And then now we have more than 300 gold standard plant genomes already sequenced. So this is the journey so far we have covered, and till now I have been involved in 15 diverse crop in which we are sequencing the genome, starting from the this rice, pigeon pea, tomato, wheat, jute, mango genome sequencing we are working currently. So this is the evolution of sequencing technology. Earlier we have the first generation sequencing, then we switched to the second generation sequencing, and now we ended up the third and fourth generation sequencing. So the major differences between these generation is the yield of the sequencing data, and also the chemistry is also pretty straightforward, and we have a less error, and we have a high throughput data. So we are switching to the high throughput data of things. But the other challenges come with the genomics is that Our sequencing is getting cheaper day by day, but what is that? The computational cost, it is increasing drastically high. So there is a new branch of science come into existence, is the computomics. It's a new omics in which the computer cost is higher. You have the sequence, but you need to have a platform where you can analyze your high throughput data. So that is the challenging part. And other than that, also we have to have a good software and skill set to analyze those data, to read out those data and extract the meaningful information. So this is the actually a key and expertise area in which people are looking forward, and they are pursuing their career. So after wet lab, the major thing is that the dry lab, like how you can interpret the data and how you can analyze the data and get the meaningful information of it. So sequencing platform is broadly categorized into two types. One is short read, the other one is long read, and now the people are working on the hybrid platform as well. Long read versus plus short read. So the hybrid. So short read was the Illumina platform, and we have a very less error in that because most of the software has already been developed for the Illumina platform. And the long read is the PacBoy and Oxford Nanopore, but we are having very few software for this. Now the people are researching, and we have like NeCat and other tools is already available for the long read. Even in the CLC, they have incorporated the long read sequencing data analysis software packages. But now the scientists are trying to do the hybrid kind of things like long read and short read. They do the long read sequencing and fill the gap with the help of the short read sequencing. So that's called as hybrid sequencing. So this is the overall step of the sequencing. First, you have a raw sequence, as I have already mentioned. Then we have a small context. We need to assemble the context, and then we can perform the polishing with the help of Recon and Pylon, in which we can fill the gap, and then finally end it with the pseudo molecule, that is the chromosome level assembly, and we can perform the annotation and further the downstream analysis. So the two assembly strategies: for the long read, we generally use the overlap layout consensus. In the short read, we have the de Bruijn graph method. So for the long read, we have a Falcon Flies software, and for the short reads, we have a spread bias. And these are the big challenges that the data analysis and the implementation, because bioinformatics is an emerging field, and data science, so in which there's a lack of expertise in this particular area. So people are sequencing, but they are not able to read the information because they don't know how to perform the analysis. So these are the things which our scientists are facing for that, because you have the sequencing data, but how you can interpret and how you can get the information is really challenging nowadays. So now come to the how genomics help in the crop improvement, as we are working on the crop improvement aspect. So we through the genomics we can harness the genetic diversity within a species and across the species. Further, we can map through the age what are the genes variants contributing to our traits and at what extent. So these are the things which we can do with the help of that. And population structure analysis, we can identify the population structure and allele migration rate and across the diverse set of panel through the genomics information. What are the marginal effect of one allele changes to the other genome? Because one genome and there is a mutation happens, so it leads to the another genome. That's how the diversity come into existence. So earlier it was only genotype, now the people incorporate genotype with phenotype. Now we are incorporating the genotype, phenotype and environment along with the microbiome. So this is the model which we are working right now, and we can trace the history of origin and domestication of different crops. I have already investigated in rice, and we have concluded that rice is a polyphyletic origin. We can develop with the help of this species specific marker, and for varietal development. Further, we can do with the genome wide association analysis by high density map, and perform the genome wide association analysis and identify the important genes and QTL reason for that, and validate that and develop a new improved variety. Further, we can use for genome information for the gene and haplotype estimation and haplotype based breeding program to assist in the breeding decision. So these are the areas in which we can employ genomics, and we can use for the variety identification and seed purity testing. Further, the thing is that using this genomic information, we can perform the functional analysis and gene editing to design a better plant. Earlier, we are just dealing with the one gene. Now, the concept is genome, and then it switched to the pangenome. And what these genome information, how we can harness this genome information and leverage to the field level, and what is the consumer preferences. So these all are the integrated omics approaches actually, and based on this we can identify the gene that we can further use for the CRISPR editing, and we can have a better plan. For example, bacterial blight resistance genes, fragrance rise, high grain number, large grain size genes. So these are the genes which we have identify through the genomics, and we can develop a transgenic as well as the edited line also through the marker assisted selection. Similarly, we have developed the powdery mildew resistance and purple tomatoes, a high oily content and linoleic acid content in the soybean, citrus canker resistance. So these are the some example of in which we can use the genomic information. So every coin has a two sides. Similarly, this the things which I was discussing earlier was all the golden side of the genomics, but still there is a lot of challenges in the genomics when you investigating the genomics. First and foremost important thing is genome complexity, because it's vary from crop to crop and in species to species. High level of heterozygosity, because there is no any such software available in the genomics that can deal with the heterozygosity problem, especially in the case of mango. When I was doing the mango, we have a huge challenge for the heterozygosity, so we have developed our in-house tool to address the heterozygosity problem in the mango, because mango is a highly heterozygous crop. Although the genome is not that big, it's a 350 around 350 MB, but the heterozygosity level is used. Degree of repeat elements, so you have to tackle with the repeated sequence and mask the repeat sequence. And genome assembly algorithm, which was designed primarily for the human and mammals, but there is no dedicated tools available for the plant genome. Poor quality genome, because people are in the running race of which one going to publish first draft, like that. So in that rat race, people are releasing the poor quality genome. So first we have to think, we have to solve all this problem, and we have to release the good quality genome, so that further the functional biologist and the biotechnologist can utilize those information for the further crop improvement. And of course, this is the high throughput science, so we of course need a funding. And also we this is a new area, so there is a lack of expertise in this field. So these are the some challenging aspects of genomics. Now I that was my general talk on the genomics and its application, and what are the challenges which we are facing in studying the genomics. So now come to my the research part, in which we have developed the unique genic 50K SNP genotyping chip. It's a high density 50K chip, and it's an indigenous chip we have developed in India, and patent for that, and it is highly useful in case of molecular breeding, and its application is rice. So I have developed both low multiplexing with high sample throughput assay, and also high multiplexing with low sample throughput assay. And both the platform have their varied applications. For instances, in case of low multiplexing with high sample throughput, we can use it for the population structure study, foreground selection, diagnostic purpose, seed quality control. And high multiplexing with low sample throughput we can use for the evolutionary study, association mapping, background selection, diversity analysis, and genomic selection. So this is the complete pipeline of the 50K SNP genotyping chip development. So we have developed this 50K genotype chip using all this pipeline, which was in-house developed. So at a time we can genotype total 96 diverse rice genotype in this chip, and this is a Affymetrix platform. And in single well, there is a 50,051 high quality marker SNP marker is present, and this was all the high assayable marker after the all QC filtering and all. So I'm not going in detail like how I have developed this chip, but it is a very good chip and with high success rate and high assayability and reproducibility is with this chip. And that chip we can use in the gene titan platform Affymetrix, and we can use for the genomic selection as a routine practice in a breeding program. So this is the percentage of gene present and the percentage of SNPs present in this chip. So you can see that the we have total four different category of gene present in this 50K chip. The first category is conserved single copy gene common in wheat and rice. Second category is single copy gene unique to rice. Third category is clone agronomically important rice gene, and fourth category is multi copy rice gene. So we keep multi copy gene as a control. So if you see that the conserved single copy gene common in wheat and rice, we have only 3700 genes, and which is 19%, but the snp is quite higher. It's a 3 to 4 fold higher here. So total 44% SNP is present. Why? Because these are the ancient genes. So the ancient gene, because these are the conserved genes, so they have the more mutations. They are more subject to the mutation, and that's why we are having the more number of SNPs. And similarly, with the clone agronomically important genes, we have 1% of genes, and similarly two threefold higher number of SNPs, because these are because these are the extensively studied gene, and we have more number of SNPs accumulated over the time. So you can see the distribution of SNPs present across the different category of the gene, and percentage of SNPs in different regions. So we have non-synonymous 20%, 41% from the intronic regions, and 7% from the 5' UTR, and 14% from the 3' UTR. And this is the physical map of rice. Rice has a 12 chromosome, and you can see the marker is throughout distributed, and it's uniformly distributed, and it's a high density SNP based array. So you can see we have captured each and every chromosome and their full length coverage is there. So on an average around we have less than we have around 6 to 7 SNPs per gene, and the distance is even less than 1 kb. So it's very high density marker. So if we talk about the chromosome wide distribution, so the highest number is coming from chromosome 1, followed by 3 and 2. And so that was the SNP array. And we have also collected and characterized more than 1000 Indian wildlife accession from different ecogeographical region of India, and developed the Indian wild rice databases, and utilize those 50K rice chip for 50K rice chip for validating this 1000 newly collected Indian wild rice accession. And we can I will show you this, that this is a single copy gene based 50K SNP chip. So we have published this paper and got lot of citation. It's more than 150 citation, and rice breeder is using it, and it's a very breeder friendly chip, and we have a patent of this chip. And the application aspect of 50K chip is that it's useful for the haplotype based phylogenetic analysis, useful in the background analysis of backcross derived mega rice variety such as Swarna Sub1, Savitri Sub1, Samba Masuri Sub1, and IR64 Sub1. Through this chip, we can able to trace the background recovery information present in their recipient parent. So you can see with the help of this 50K chip, we are able to deduce that the Swarna Sub1 has a highest genome recovery from the recipient parent is 97.7%, followed by the 91.9% in the Savitri and IR64, and the lowest in the Samba Masuri Sub1. So because of this chip, we are able to deduce that the Samba Masuri Sub1 required more back crossing from the recipient parent to get their genotype. Genomic information. So these are the things. And haplotype based phylogenetic analysis we have performed in the diverse 128 rice genotypes, which including wild type, aus type, indica, japonica type, and they were beautifully stratified into four different groups. Group 1 is entirely wild type, group 2 is aus type, group 3 is indica type, and group 4 is japonica type. So the haplotype based phylogenetic analysis is clear cut distinguish between all these four groups. So this is the different between the general phylogenetic we are doing. This is the actually gene based haplotype analysis. So there's a very novel concept which we did on this. So apart from that, there are several other applications. For instances, we can use for the 50K chip for the phylogenetic analysis, QTL mapping, evolutionary study, background analysis, and genotyping for the large number of sample. And of course, it acts as an excellent biomarker to tag any gene. So the property of the 50K chip, it's a single copy gene based design. So it has a non-redundant high quality SNP probe, high sample success rate, high assay reproducibility, high genotyping call rate, and on an average around 6 to seven SNPs present per gene, and it has a less chance of cross hybridization. It is quite cost effective because at a time we can genotype total 96 samples. So novelty of the and significance of this work is this is the first report of genic rice SNP genotyping chip, and it has a high success rate. And the rice community and rice breeder and researcher is using it extensively, and they have developed several variety. Even from our group, we have developed four variety using this 50K chip. So it is proven as an efficient and reliable tool for the Indian wild rice germplasm characterization, genetic background selection, association mapping, QTL mapping of important agronomically quality trait, diversity analysis, and develop the QTL map for the salt tolerant by the bulk segregant analysis and recombinant inbred line. And two submergence tolerant rice variety were developed, such as Samba Masuri such as Ranjit Savan and Bahaduran Savan. And the most important aspect of this chip, it help in reduction in breeding time, because the conventional plant breeding method based on the rigorous phenotypic selection on it's depend upon the breeder experience to develop a new and improved variety. But because of this chip, this most of the things we can short out at the molecular level, and we can reduce the generation time. So we have a high genetic gain. So on an average 3 to 5 years will require to develop a new improved variety, but earlier it was 10 to 15 years. So you can see we have a 10-year reduction in time, so we can have multiple variety with the help of such important high throughput genotyping platforms. So this is the beauty, this is the major application of this chip. Now come to the other aspect that that I was working on, that the low density 48 plex Elimla golden gate assay for the characterization of Indian wild rice germplasm for their population structure analysis. Because for the population structure analysis, we need genome wide unlinked marker so that it can cover the entire length of the chromosome, and based on that we have the three different population sizes. One is the progenitor of indica type, progenitor of aus type, and the third one is mid gangetic plane. And you can see this is the phylogenetic analysis of 418 Indian wild rice accession, which we have collected and characterized both morphophysiologically as well as the genetic level using the markers. And also try to address the question of origin and domestication of Indian wild rice and their relationship with the cultivated rice, because our war is China is not only restricted to the boundaries, because they are always claiming that this particular crop or this particular species has been originated from the China, and they have given throughout the world. But that was not completely true. So we just try to reinvestigate this thing, because there's a two hypothesis, rice origin and domestication. One is monophyletic, the other one is polyphyletic. So the monophyletic was almost 2 decades ago, and they started researching, and they have a model that a rice has a single origin model. It means Indica and Japonica were domesticated in the same region from the wild ancestor, but that was not true. And they claim that the Indica and Japonica were originated from the same ancestor from the Yangtze River of the China. But later on, the other groups independently they have investigated that rice is a independent domestication, and Indica and Japonica were domesticated separately, and it has a different geographical path. Okay, so and my group as well as other group independently, they have investigated based on the some key agronomically important domestication related genes. So during my study, I have shorted out total 13 key agronomically important domestication related genes, which is present in the wild type but absent in the cultivated rice. For example, the pericarp color, you can see that the pericarp of wild rice is different, and the cultivated one is entirely different. Similarly with the hull color, grain sizes, amylose and amylopectin content. So these are the some domestication related traits which we can easily distinguish and perform the haplotype based analysis. So this is some morphological variability in Indian Oryza rufipogon complexes. So you can see that the extent of awning, internode pigmentation, hull color, and their their height, and also the grain sizes. So these are the different characteristics of the wild type which is absent in the cultivated type. So based on this, we have investigated the evolutionary insights of this domestication gene, and through this we concluded that rice has a polyphyletic origin. For example, the haplotype based phylogenetic analysis, in which they show biphyletic origin, in which the H3 is the ancestral haplotype which is having all the pericarpus black color, followed by the H4 we have all the pericarp black, followed by brown and little bit green. And in H2 we have a house type along with the wild type, in which black, green and white. But H1 is the recently evolved, and it's an independent region which is all the cultivated type. For instances, we are having the aromatic type, japonica type and indica type. So all three are nested here in the H1 haplotype. Similar sort of analysis I have performed in the different category of gene. For example, grain size, because grain size is also a domestication related genes. And now we are, for example, in basmati we have a long grain and other things. So GBS it's again a star synthase gene, because japonica rice is more kind of sticky and indica rice is not that sticky because of this amylose and amylopectin content. So these are the different form of amylose and amylopectins are investigated the study. So the summary of this study is that the Aus and Indica group of rice cultivar were domesticated in the Indian subcontinent, where they have their closest wild relatives. So they have originated from the wild type of their native countries, and resulting supporting the polyphyletic origin of cultivated rice. And mid Gangetic plain, upper Gangetic plain and eastern hill region of India are the source of Indian rice gene pool. So diversity within the wild rice is of immense value for rice improvement, especially for impending climate condition. And we can utilize this wild rice resources, and we can meet the demand of rising population, and we can develop a climate resilient variety. So that is the goal of harnessing the genomic information from the wild type using the functional genomics tool. And now switch to the genome wide association analysis. Since we have this high throughput markers, high throughput genotyping platform, so we can perform the genome wide association analysis. Also we can perform the transcriptome wide association analysis, and genome proteome wide association analysis, genome phenome and environment wide association analysis. So see, in this line we have developed two variety and registered into the NBPGR. And other than that, the genome sequencing I have been working on, I'm still working on the mango genome sequencing project. You know that the mango is native to India, and we have sequenced first the Amrapali genome, and then later we have sequenced the Neelam and the Sherry. So it's a novel concept based on the trials binning assembly approach. And heterozygosity is a major problem in this, and also develop the 82K SNP chip for this same concept as we have done in the rice, and sequence total 384 diverse mango genotypes using this 82K platform. So this is one interesting study in which we have done the mango gene conservation in the 29 species, and this is a two-way diagram in which the mango has total 39,150 genes, and out of which 3,825 genes are unique to mango only, and rest are sharing with the across 29 diverse species. And we have also studied the evolution of how the mango is, and then perform the this chip development. And utilizing this chip perform the genome wide association analysis of agronomically important and pomologically important trait. For example, fruit length, fruit weight, blush intensity, polyembryonic alternate bearing in the both Neelam and the Sherry. And two multi-location data is there, one from the IRI New Delhi, the other one is from the IHR Bangalore. And I've been also involved in this the wheat 35K wheat breeding Affymetrix, in which we have just trying to identify the low and high phosphorus related genes present in the wheat. And wheat is a hexaploid, and this wheat is a tetraploid. And also developed the first generation haplotype map, hap map of T. Here we have analyzed the data of 369 diverse T genotype, and it was the complete pipeline developed by me. And we have also database developed, and you can see that the T is a used genome. It has a 4 GB genome size, so 4 GB into 369. So how you can imagine the data point in this. So but we have able to successfully concluded this, and this is a first report from the India as well. So in line of that, also this is my DST inspired grant in which I am working currently, and about to finish. Almost all the objectives are almost finished, and we got very interesting result. Develop the 62K genic arhar chip, and you can see these are the diverse arhar type. This is a tour dal actually, and we have a collection of more than 400 diverse genotypes of this pigeon pea. And first perform the screening, phenotyping screening, and also the genotyping using the 62K chip. So identification of novel gene and QTL for seed quality and nutrition trait was the major goal of this project, in which we are just trying to looking the high protein line and the high protein content line in the pigeon pea, and what are the gene responsible for the high protein content in the pigeon pea. So first for that we have screened the protein lines using different method, Geldal method, and also the Dumas nitrogen analyzers, and then perform the association analysis. So this is the overall methods. First we perform the genotyping and phenotyping of the different traits we have mapped, then perform the genome wide association analysis and using different models, and then identify that the mixed linear model is working really well in this case, and then identify the, because first you need to run the different models and identify based on the crop type which model is really working well, and then identify the causal gene responsible for that. And after identification of the causal gene, you can perform the functional analysis. So these are the some pictures of the high protein line and the plant type and plant height. You can see we have a pod length variability in the pod length and the number of pods, also the pod color, flower colors. You can see we have a very yellow, so dark red purple color flowers in this. Also it's a use data, because we have all the genotyping data and a use phenotyping data. To date we have collected 27 different qualitative and quantitative data of pigeon pea. So develop a database in which we have all genomics, phenomics, transcriptomics, metabolomics database related to the pigeon pea. So this database is dedicated to pigeon pea only. The name is pigeon pea omics databases, and paper is already accepted of this database. So in this work we have mapped various phenotypic traits, quality traits like flower color, flower streak pattern, sheet coat color, sheet shape, leaf shape, and quantitative traits including plant height, pod number, pod length, pod width, seed weight, stem width, and leaf angle, branch angle, and so on. And screening, after screening the high and low protein line, we just wanted to make a cross and make a intermediate line of some desirable traits. So this is the crossing which I am performing in the field as well, and also trying to investigate the sterilitic mosaic virus. This is a biotic part of this work. The sterilitic, because pigeon pea is prone to the sterilitic mosaic virus and also the port borer. So and these are the major problems in this particular crop. So we are also trying to address at the proteomics and metabolomics level of this particular crop, and develop the high density linkage map of the pigeon pea, because the high density linkage map is really lacking in the pigeon pea. And again perform the genome wide association analysis for this using the this genotyping information and phenotypic information. We have integrated it, and you can see this is the seed weight. We have a significant SNP present here. So out of 62,000 marker we have, for example, in case of seed weight, 5 markers is really significant. So these 5 marker we can screen for further downstream analysis, for the validation, we can say that. Similarly, with the several different traits, we have performed the similar sort of analysis. For example, seed length, seed weight, and the most important is total protein content, in which I was interested. That I was interested in. So we have, you can see, we have find huge number of association, number of SNPs present in this. So these are the some traits which we have mined, and you can see the annotations and the locus and all the detailed information is present and plotted on the chromosome level as well. And this is the patient omics databases, and this is the complete pipeline which we have gathered all the information related to that. And also we are looking for the complete proteome atlas of pigeon pea from the different tissue specific at the different developmental stage, so that we can have a complete proteome map of that particular crop. So because it's not there, and it's a not a western world crop, so we as an Indian we have to invest more on such crops, or it's a South African and other people is working on that. So I have a collection of total 13 different countries, so that we can utilize it a very great level, because all the crops are entirely different, and it has a huge diversity that we can see with our eyes while growing in the field. So during that journey, also identify that the DOF is really very interesting factor, that is DNA binding protein present in that, and it is actually having a crosstalk between the salinity and drought. So we have also identified what are the reason and domains which are really contributing that, because pigeon pea is a drought tolerant crop. It doesn't require much water. It can grow in a marginal water in a very less input, and also it's a good carbon sequester, so it has a good carbon capture capacity. So pigeon pea is a multi-purpose crop and also a medicinal value. We can use it as a nutritional and nutraceutical purpose both. So these are the some bioinformatics analysis which I have conducted, and also looking for the sterilitic mosaic virus 1 and 2 in pigeon pea using the proteomics aspect of the different ligands. And since we have gathered so much of information related to the omics, so the my major goal is to do the proteogenomics approaches and integrated omics approaches, and deployed into the machine learning and AI based approaches, so that by integrating all the genomics, transcriptomics, proteomics, metabolomics, and anomics information, we can pinpoint what are the genes which is constantly present across that, and a key player for this, and which we can go for further utilize for the editing purpose or marker assisted selection, and the other purpose to make the crop improvement and use for the breeding program. So this is my broad research vision, that I wanted to work on the yield aspect, genotype by environment interaction, carbon capture, and also one of my student, he is interested in pea phosphorus use efficiency in pigeon pea. So we have also found very interesting result in that. And also my area is nutrigenomics, because I want to work on development of biofortified crops. So this is my broader research vision and this. And these are some new leads which we are working on the future goals: to develop cost effective high throughput genotyping technology for genetics and molecular breeding application in diverse crop, identification of novel genes and QTL for seed quality and nutrition traits in pigeon pea using GWAS approach, development of genomics and proteomics tools and databases for different crops, including pigeon pea as well, and also develop the genome assembly association pipeline for the long read sequencing, because we are having now the very good long read sequencing platform, but the paucity is a good tool. So we need to develop a more tool so that we can easily smoothly conduct the data analysis. And of course I'm more interested in that the nutrition and food security aspect, because that is really needed since I'm working on the plant. So people are shifting toward the plant based diet, and especially the vegetarian peoples are relying on legumes for their meat, to take as a, they consider it as a meat, and get the nutrition of that. So these are the some new leads and the future goals of my work. So throughout my journey, I have received many awards and accolades, including in some Medal of Young Scientist and NASI. And recently, I have been inducted as an NEAS Fellow in the ninth AGM meeting. And these are the some teaching and extension activities I have conducted and guided students uh, till now, and also doing a lot of extension activity, hands-on training on NGS things, and also the tool. So this is all, and this is my all research summary uh, till date so far. And thank you all, those who have, of course I want to acknowledge my mentor, my students, and my funding agency for supporting me to conduct this work. And thank you all for your patient hearing me. And I will be happy to take questions if you have any. So thank you so much.
So, thank you so much, ma'am, for the extremely knowledgeable and interesting session. Your work in the field of bioinformatics is incredibly remarkable, and I'm pretty sure you've inspired all of us to seek more knowledge in this field. So, it's time to take some questions now, and please post them. So, ma'am, we have our first question, which is how to save the germ plasma of the seeds for genetic conservation.
Yeah, great question. So for that, you have different agency. For example, in ICR, we have for the germplasm uh, preservation, that is NBPGR, National Bureau of Plant Genomic Resources, in which they are taking all the genomic resources. So you have to give all the passport detail. They will and they will provide you the form that you need to fill, and then they register. And also they will test that the germplasm which you have collected is what is that and from where it is. So you can collect there, and if you are collecting from your own side, so you can manage for some time at your institute, and but later on you have to register somewhere, then only it is known as that registered. They will provide some ICP number or some number, that is ICP number, so that it is registered, because you have to register your germplasm in any agency. In ICR, we have NBPGR. Yeah.
The next question is how to address the genetic changes in crops.
How to address the genetic, in what aspect you are looking for, that what genetic changes you want to see. So, for example, you want to see the genetic changes. So first, what you are observing through the phenotyping, right? Because first is things is where what the plant is appear for, and if it is uh, within a species, if it is drastical change, so what you can do, you can take a sample and perform the sequencing, and then you can compare the base pair by base pair through the alignment, what are the changes. Because if we talk about the humans also, like in case of human being, we are 99.9% similar. 0.1% variations is because of the single nucleotide polymorphisms, right? So you can do the synteny analysis, in which you can identify the similarity and what are the unique features that you can also stimulate. So there is a software also to estimate the diversity through the comparison, because if you are comparing two species, so you can sequence the two species and perform the alignment, and you can capture the variation through that at genetic level. And if you do multiple also, then you have a different platforms like GATK pipeline, in which you can perform the variation. You can capture the variation across like 1000 or different number of genotypes which you want. So it's again depend upon the how many accessions you have, how many germplasm you want to look. If you want to compare between two, it's a comparative genomics. If you want to look other, it's a pan genome approach.
We have a next question, which is how functional genomics helps in crop improvement.
Oh, wonderful question, Renuka. So, how functional genomics help in crop improvement? So, for example, we do all the bioinformatics analysis, and we have traced that these are the SNPs, or these are the genes which is responsible for example, drought resistance or drought tolerance. So, you know that DREP gene is responsible for this particular crop is drought tolerant. So, you can identify through functional genomics different tools, and then you can identify the genes, and you can do the validation through RT-PCR cloning, and you can raise the some transgenic also of which is having the drought resistant genes. So these are the things which you can do with the help of the functional genomics tool, because these are the tools you need to use. How can NGS and high throughput genotyping contribute to accelerate the development of climate resilient crop? So NGS, as you know that earlier we have that platform, first generation sequencing. So you can see that the human genome or itself take 10 years to have a sequence, right? And now last year only we have a gapless human genome. So the power of NGS is that it reduces your time. It also reduces your cost. Similarly, with the high throughput genotyping platform, it reduces your time and cost to genotype, to have a markers, to have a data, so that you can mine those information, and those information you can deploy for the crop improvement. For example, let's say if a breeder was doing crossing by the traditional method, but it was based on the traditional way. But in the genotyping, the molecular way is that the field time it get reduced. You have to go field, but first you can sort out many aspects through your lab level. So that is the power of the NGS. Okay, so you can see you have a data within a 1 or 2 days of the sequencing, right? The later part is just to analysis.
We have another question, which is what challenges and opportunities exist in managing and analyzing the vast amounts of genomic data generated by NGS and high throughput genotyping platforms?
Okay, so the opportunities first, I will talk about the challenges, then opportunity. So the challenges is that the NGS data, as you know, that the next generation sequencing data and high throughput. When the word is high throughput, it means a huge data set, right? So first thing is you need a computational power, okay, where you can store the data. Second thing is you have an idea that which tool and software is really good to conduct such experiment, and you have a sound knowledge of bioinformatics, coding, computational, and along with the biology, because you are dealing with the biological system. You are reading the data, but still you have to connect the dot of biology. So you are interpreting, but how it's related. So that's how the analysis came. So these are the some key challenges that people, because some people are good in coding, but they are lacking the biological information. They are not a biologist, because for example, the deep computational biologists, those who are working on the development of algorithm, but they need one biologist that what are exactly they are looking for, right? So that is the, if you have a blend of all these component, then this is actually very rare. And also the computational system is required, a use system is required. Now these days we are having the online Amazon clouds and all those things, so you have to know, you have a skill to work on the cloud computing, right? Because we are entering into the era of AI and ML. So you have to have everything is on data. Well, even scientists is just plucking their sample and they send to for the sequencing, and then later on we are dealing with the data. So you have to have a very good analytical data skill set. You know how to work in the Linux environment. Coding knowledge is essential. So these are the some challenges which people are still learning and biologists still lacking. And if this is the challenges, then the same time it is an opportunity for you to explore it. So if you are good in all these things, so you can develop a good tool and algorithm for the long read sequencing, which we are lacking right now. And also you can train other manpower in this direction, because we are very few people, those who are doing this kind of work. And you can reduce the time of the analysis, and also the time of the crop, because if you do anything on the computation non-computational way, it take a longer time. What is the benefit of having computed, that it reduces the time and enhance your efficiency. So that is the opportunity for you, so you take that challenge as an opportunity same time. Any other question?
Okay. We are done, ma'am, with all of the questions.
Okay.
So, yeah, thank you so much, ma'am, once again for giving your time and for the great interaction. It was a pleasure to have you here with us today. So, it's time to felicitate our speaker of the day with a memento and a certificate of appreciation.
So thank you, Simran, for having me. And I believe that everybody has gained some insight on this, and I understand that this is an emerging field. So people, but at the same time, it is a potential field for everyone to explore, because if there is a challenges, then there is an opportunity. वो कहते हैं ना, आवश्यकता ही आविष्कार की जननी है. So if there is a need, then you will do. So yeah, thank you so much for having me on this and this special occasion. So thank you so much.
Thank you so much, ma'am.
So may I log out now?